Thanks so much for coming to the first installment of Wind Down with Science, presented by UW Medicine. My name is Lonnie Swingle, and I'm the Sponsorship and Events Manager here at UW Medicine. And I'm really excited to launch this series here today. This South Lake Union area is so dynamic and so vibrant. It really mirrors a lot of the inspiring work that's happening here at UW Medicine, so I'm happy to share that with you all. So how many of you in here are true crime fans? Yeah, okay, good. I thought there might be a few here. I think you're gonna be fascinated by this talk, and I'm sure you'll have a lot of questions as we go, but I'm gonna ask that you hold your questions until the very end of the session. Everyone will have a chance to ask any questions of our wonderful speakers here today. We're honored to have two amazing speakers with us to answer the question, can your DNA solve a murder? You'll hear first from Dr. Celeste Berg, Professor of Genome Sciences at the University of Washington. She earned her PhD in molecular biophysics and biochemistry at Yale University, but has focused her scientific research studies on understanding the genetic mechanisms that regulate development. Dr. Berg also enjoys teaching, and in an effort to attract the best and the brightest to the field of genome sciences, she collaborated with Dr. Bonnie Brewer in the Genome Sciences Department to develop a hands-on forensics course for UW freshmen. This course, called CSI Seattle, introduces students to the methods and logic of forensic analysis by having students investigate a murder set up by the UW police. In addition to research and teaching, Dr. Berg has served on numerous national and international advisory boards. She's currently a member of the NIH National Advisory General Medical Sciences Council. Then we'll hear from Dr. Stephanie Malia Fullerton, an associate professor of bioethics and humanities at the University of Washington School of Medicine. She received a PhD in human population genetics from the University of Oxford and later retrained in ethical, legal, and social implications research with a fellowship from the NIH National Human Genome Research Institute. Dr. Fullerton's work focuses on the ethical and social implications of genomic research and its equitable and safe translation for clinical and public health benefit. When Dr. Fullerton is not leading the Bioethics Advisory Board of the Kaiser Permanente National Research Bank, you might find her belting out a tune at karaoke. And now it's my privilege to introduce Celeste Berg. Oh, yeah.
It was a cold, dark night in November 1982 when the police received a phone call. They investigated and found the body of a young girl. She'd been raped and strangled with a tie from a bathrobe. Her body had been left in a cardboard box behind a TV repair station in Magnolia. Further investigation revealed the victim to be Kristen Sumstad. She was only 13 years old. Now imagine that you are one of the detectives solving this crime. What would you do to solve this murder? Keep in mind it's 1982 and you don't have the tools that we have now. What would you do?
Fingerprints.
Fingerprints, great. Fingerprints, what else?
Blood type.
Blood type?
You said it was a rape, so maybe check for like any DNA samples left?
1982, no DNA. But interview family members, great. Other ideas?
Tire tracks around where she was dumped.
Tire tracks, other sort of physical evidence. This is great. These are all the things that the police did at that time. They interviewed neighbors, they looked for fingerprints, they tried to track down that bathrobe tie that she was strangled with. They essentially did everything they could to try to look for suspects. And through their efforts, they were able to eliminate a number of individuals who had really good alibis. But unfortunately, they did not have enough evidence. There wasn't sufficient evidence to arrest anyone. And as a result, the case went cold. And it sat, all the evidence from that crime sat in the police crime scene lockers for 10 years. And then the cold case team brought the evidence back out again and re-examined it. And they thought it was worthwhile to do so because in the intervening decade, there had been a paper published by a group in Britain and demonstrating that it was possible to use DNA for analyses in forensics types of investigations. So the police tried to purify some DNA from that rape kit sample, but unfortunately they did not have enough DNA using the methods back from the late 80s and early 90s to be useful for this particular type of test. So the case went back into storage until 10 years later, the cold case team brought the evidence back out again. And in the meantime, there was another new invention, another method that had been developed that let investigators take small amounts of DNA and amplify it for analyses. And sure enough, when the detectives analyzed that sample with those methods, which I'm going to talk a lot more about later, they did find really high quality DNA. But unfortunately, when they compared that DNA to what was available in the FBI or other police databases, there weren't any matches. At that time, all the DNA in those databases were from convicted felons. But the police had this DNA now, and so they looked really carefully back through all their notes from the time, and they came to suspect one particular individual. There was a young man named John Athan, 14 years old at the time, who had been seen pushing a large cardboard box down the street. And when questioned by the police, he claimed that the box held firewood that he'd stolen from a neighbor. And they couldn't prove otherwise, and so they let him go. But they suspected he might be the culprit because he had been hanging around with the Sumstad family and sort of pestering the sisters quite a bit. Now, the police don't have a great budget, and by this time, John Athan was married and living in New Jersey, ran a construction business, but the police couldn't afford to send somebody out to New Jersey, so they tried to think of a really good way to get a sample of John Athan's DNA, but do so inexpensively. And they came up with a really incredible ruse. They pretended they were lawyers, and they created a class action lawsuit against the city of Seattle, claiming that people had been overcharged for parking fines. And they knew that Athan had received a few parking tickets, and so they created this class action lawsuit. Now, there were a number of features about this lawsuit that were really important. First of all, the supposed law firm really consisted of the names of all the police department staff. These are their real names. And on top of that, the address was the police department address. Okay. They sent off the letter, and for the price of a stamp, which at that time was 37 cents, sent it to New Jersey to one person in this class action suit. And John Athan fell for it. He filled out the forms, put it in the envelope, licked the flap, sent it back off. And Dr. Beverly Himmick, who at that time was director of the Washington State Crime Lab, she was able to obtain sufficient DNA from the saliva on the flap of that envelope to match it to the crime scene. 21 years after Kristen's death, her killer was convicted of murder. Now, John Athan had a really good lawyer. He appealed the case and claimed that the police's ruse had trapped his client. The case went all the way to the Supreme Court in Washington State, but they ruled in favor of the police, saying that the police often create ruses when they're trying to capture prostitutes or drug dealers or something like that. And they had used their real names, real addresses. If Athan had even done a little homework, he might have figured this out. But he voluntarily submitted his sample. In the end, Athan served about 8 years for a crime that he committed when he was 14, which is a little bit longer than most youngsters serve for a crime of that type when they're 14. But it was the DNA analysis that eventually captured the killer of Kristen Sumstad. So today what I'm going to do is talk a little bit more about the methods that scientists use to solve these types of cold cases. I'll spend most of my time doing that, and at the end, I'll talk about some new methods that are just now being put into play that are raising a lot of ethical issues, and it'll set the stage for Dr. Fullerton's talk. Before I go into those methods, though, I want to point out that DNA is useful not just for capturing the bad guys. It's really important in identifying victims, say in a plane crash or natural disasters of some kind, and equally, if not more important, to clear people who are actually innocent. In fact, the Innocence Project makes its goal the use of DNA to free people who are wrongly convicted. So at the beginning of today's talk, we came up with some list of ideas of how the police should go about identifying a suspect for a killer. And the police still use all these methods. It's not that those methods are bad. It's just that now, whenever possible, investigators try to get DNA to analyze. So why do they do that? Why is DNA analysis so powerful? Well, there's actually 3 real reasons. The first is that it's definitive, and we'll see why in more detail now, but let me just set this up. All of us have DNA that's virtually identical, 99.9% identical, and that's what makes us human instead of a chimpanzee or a gorilla or something like that. Our DNA is very similar. But that 0.1%, when you think about 3 billion base pairs, is enough to make us different. Let's take a look at the DNA and how it differs. So you know that each of us has a set of chromosomes, one from mom and one from dad. And if we were to look at a chromosome and pull it all out and unwind it, we would see a DNA double helix like this. Okay? In 2001, the human genome sequence was published. It's actually called the reference genome. It's actually a composite of DNAs from a lot of different individuals. But since 2001, there has been thousands of DNA sequences from other individuals revealing differences in our DNA. And those differences, for today's purposes, fall into 2 main types. The first has to do with these short little repeat sequences, CA, 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 or other sorts of little short repeats called short tandem repeats. And they vary tremendously amongst the population, far more than the rest of the DNA. The other type of difference is called a single nucleotide polymorphism, poly-many morphism shape. And that type of difference comes into play at the end of my talk, and that will be the type that's used for this other type of analysis. So the differences between us are definitive. That's one reason why DNA analysis is so powerful. But another reason it's powerful is that DNA is quite stable. For example, about 10 years ago, forensic scientists investigated a grave in the Ekaterinburg forest of Russia, a grave they suspected contained the bones of the Tsar Nicholas and his wife Alexandra and their family. The Romanov family had been killed by the Bolsheviks in 1918, and their bones buried in the woods, their bodies buried in the woods. taking DNA from those bones and comparing it to DNA of living ancestors revealed that indeed the forensic scientists had found the correct remains. That's 100-year-old DNA. But the technology is so good now, it is actually possible to analyze DNA from our long-distant relatives, Neanderthals, from bones that are 30,000 years old. And in fact, a number of groups have sequenced the entire genome of several different Neanderthals, as well as Denisovans and other of our ancient relatives. So the DNA is stable. That's a great feature. The third reason why DNA is so powerful is that the methods available now are really quite sensitive. A single cell, a single hair if it has the follicle, because that's where the DNA is, a single skin cell, a single sperm cell is sufficient to analyze. That's incredibly powerful. So now let's take a look at the methods themselves and see why they help reveal the differences between people and why a single cell is good enough. So I told you that the main method used is to analyze what are called these short tandem repeats. Most of the repeats that scientists investigate are 4 nucleotides long, and they vary greatly between individuals. Oftentimes, the alleles or differences or variants between people are categorized according just to the number of the repeats that a person has. The FBI and other forensic scientists investigate or characterize 20 different sites in the genome that have these short little repeats that vary quite a bit. Initially, they started out with these 13, but since January of 2017, they added 7 more sites in the genome so that they could compare DNA from our samples to DNA obtained in Europe or Asia or other laboratories that were using somewhat different variable regions in the genome. And now everybody's using these same 20 sites so that the DNA can be compared all over the world. What do the data look like? Well, what people are actually trying to do is compare fragments of DNA that are slightly different sizes. So what you're seeing here is 3 different loci from an individual, a single person, and the different fragments from chromosome from mom or chromosome from dad are slightly different from each other. They vary in the repeat number. There's 14 repeats or 15 repeats at this 3rd chromosome locus, or 17 repeats and 18 repeats at this VWA locus, or 23 and 24, and so forth. Okay? Now, if we look at another individual and perhaps evidence from a crime scene, we would see that this individual, this suspect, has 15 and 18, just like the crime scene, 17 and 19, 23.2, 24, as opposed to the suspect 1, who had these different sizes of fragments. Okay? All right, so we looked at 3 loci. Why look at 20? In fact, why is it important to even just look at more than 1? Mm-hmm.
A sample of DNA might have been damaged in a certain spot, and so you might not be able to get to a certain loci, so you need to have multiple locations to be able to check.
That's a very important point. I'm not going to talk at all really about some of the problems with DNA analysis, but I think Malia will. But you're right, that is a possible problem, especially if you have very little DNA. Other ideas?
If you're just looking at 1, there'd probably be a lot higher possibility that they're the same.
They're the same, but why might they be the same? Why might they be the same?
They're relatives.
They're relatives. Certainly if they're relatives, that's a very high chance.
There's only so many possible combinations.
Oh, very good. There's only so many possible combinations. And so it's actually possible that several people might have the exact same combinations of alleles just by chance. And it just depends on the frequency of those alleles, those variants in a particular population. So if you're only looking at one locus or even just 3 loci, a lot of people might match. But let's take a look at what happens if we now look at, say, 13 loci. Now, if we look at 13 loci and all of them are matching these particular alleles from the crime scene, the probability of that, based on the frequency of particular alleles in the population, is actually 1 in 4 quadrillion, more than the number of people on Earth. And that's just with these 13 loci. When you look at 20 loci, the probability of finding a match is something on the order of 1 in 60 quadrillion. And that's what I mean by, if you're getting a match of this type, it's definitive. It's the same person. The only way you could be wrong is if the suspect had an identical twin, because identical twins' DNA is the same. All right. Okay, so how do we obtain sufficient DNA? I told you that a single hair cell or a single sperm cell was enough. Let's take a look at this method. What we actually do is make a lot of copies of just those regions in the genome that we care about. So we obtain DNA from a crime scene, we characterize it, and we use a little trick. It's essentially photocopying just the region of DNA that we care about. So let's think about how this works. What we have is a book that contains a region we're interested in, and the book would be the whole genome. And we want to make copies of just this particular quote from the book. So we need to somehow identify that quote in the book, and so we're going to put little post-it notes around the region that we want to make a lot of copies of. And then we're going to essentially Xerox just the little part that's post-it note flanked, okay? So the book that contains the quote is the entire genomic DNA from the sample. And the post-it notes are what we call little DNA primers that will flank the region that's really variable. But those post-it notes are highly conserved amongst all people and shared so that it doesn't matter whose DNA you're analyzing, they're always going to find the quote. Okay? And then we need some raw materials for making the copies, and we need an enzyme that can actually make the copies. So in the next few slides, I'm going to use this diagram to indicate double-stranded DNA as genomic DNA. I'll use this diagram to indicate little single-stranded pieces of DNA that are the post-it notes, and then just A's, G's, C's, and T's to indicate the raw materials, and a circle to indicate the enzyme that's actually making the copies. Now this method, the polymerase chain reaction, was invented by a man named Kary Mullis, who received the Nobel Prize for this invention back in 1993. And his idea truly has revolutionized DNA analysis. So let's take a look at how the method works. We start off with these 4 components, our genomic DNA, it's also called a template, our post-it note primers, our raw materials, and the enzyme that will make the copies. And the first step is to denature the DNA, to heat it up really high. And the double-stranded molecule comes apart and separates into 2 single strands. You then lower the temperature a little bit, and it allows these post-it note primers to find sequences that it can form what are called hydrogen bonds. T's pairing with A's, G's pairing with C's, and where they find a match, they bind. Then you raise the temperature slightly to the temperature that the polymerase really likes a whole lot, and it begins to synthesize DNA. At the end of the first cycle, it's a polymerase chain reaction, you have 2 copies of DNA where you only had 1. But you continue the cycle, say 30 times, and it works in part because even though you might start with only a single DNA molecule, everything else is in vast excess, and so you keep making more and more and more. 2, 4, 8, 16, 32, 64, and so forth, so that after just a couple of hours, you have billions of copies of just those little regions. Part of the reason this works is that the enzyme that we use, normally proteins or enzymes, they would just completely fall apart if you heated them up. That's what happens when you fry an egg. You heat up the protein in there. But this enzyme comes from literally Yellowstone, and it likes the heat. So it can stand these cycles. Okay.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
And the length of those molecules will vary depending on how many repeats a person has. So we've talked about these methods. In the last few minutes, what I'd like to do is turn to the use of a new method that doesn't analyze those short tandem repeats, but instead uses the differences of those single nucleotides throughout our genome, coupled with analyzing genealogies to try to track down killers. And this method is so new and it raises so many ethical issues. I'm sure there will be a lot of debate about this topic in the future because more and more forensic investigators will turn to it. The Golden State Killer eluded the police for over 40 years for a number of reasons. Part of the reason that he was so successful at eluding the police was that his modus operandi changed. He started with burglaries, he then began raping women, and those eventually led to homicides. He also moved in California. He started in Sacramento and moved to the Bay Area and then down to Southern California. He also was pretty savvy. He picked his victims knowing that there was a good escape route, a park nearby, a bike trail, someplace he could get away quickly. He also wore a mask and left no fingerprints. It was much later when DNA from homicides from Southern California were connected to homicides here that FBI and other investigators started considering the possibility that a single individual, not 3 separate people, had carried out these attacks. And much later, the rapes were finally tied into the same person. Now, one of the investigators who'd been trying to track down the Golden State Killer was Paul Holes. And about 9 months before he was supposed to retire, he decided he was really going to try to find this guy. He had heard from another detective that it was possible to use genealogical databases to identify individuals from a DNA sample. And in fact, this other detective had identified a kidnap victim that way. And so Paul Holes determined to do the same sort of thing. And he worked with the FBI and a pathologist from Irvine to obtain DNA from a rape kit. And instead of analyzing it at these 20 STR loci, the way people do, he analyzed the DNA the way that 23andMe or Ancestry.com might analyze the DNA, looking at those single nucleotide differences. He then uploaded the data into a database called GEDmatch. He chose this database because unlike 23andMe and Ancestry.com, this database actually allows the police to search the database. Okay? And they found a match to distant relatives, great-great-great-grandparents of the suspect. Holes and a team of 5 investigators spent many months putting together 25 different trees with thousands of individuals in them. And eventually they came down to the future, looking at census data and obituaries and police records, all kinds of genealogical methods. But eventually they came down to the present, and they had 2 suspects that were attractive, in part due to their age and the fact that they lived in the Sacramento area. One of those individuals they eliminated with DNA evidence. But the other one led them to a disgraced police officer who had been fired for stealing from a hardware store. Okay. They followed him, and they were able to obtain DNA from his car handle. And when that matched their suspect sample, they went back a week later and obtained a tissue from his garbage can and tested that again, and that matched also. And when that second match came in, the FBI and Paul Holes and others arrested Joseph DeAngelo as the Golden State Killer. Now, this genealogical testing is possible in part because there's so many people who have put their DNA into databases, maybe some of you. And we'll hear more from Malia about how that could be potentially a problem. So what I've told you about today is how DNA evidence is really handy at catching the bad guys, clearing the good guys, identifying victims. We talked a lot about the methods, including a new method that has implications for the future. I have here some websites with a lot of information about the various cases I've talked about, and I want to give special thanks to Bonnie Brewer, my partner in crime, who helped me develop the CSI class. And without further ado, I'll hand over the mic to Malia.
All right. Thank you so much, Celeste. Really great lead-in. Good evening, everyone. Pleased to have you here. I'm now that we've heard a little bit about the science and how effective some of this is, we're going to talk a little bit and very quickly about some of the possible ethical and social concerns that might accompany the uses of forensic genetic testing. So very briefly in my time this afternoon, I'm going to be spending a little bit of time talking about the way that we can kind of conceptualize or think about possible ethical concerns in the context of basically balancing competing interests, balancing the need to kind of basically ensure the common good and ensure public protection via identifying criminals versus protecting individual interests in privacy and autonomy, for example. And how do we think about that, and what are the ethical concerns that are raised by these kinds of technologies? In order to kind of introduce or think more specifically about some of those concerns, I'll spend a little bit of time kind of giving us a brief historical take on where forensic genetic testing started and where it's going. And we already got some a very nice kind of prelude of what's now possible with regard to catching the Golden State Killer, and then basically end by thinking about what it might mean now with so many of us, probably including many people in the room, engaging in direct-to-consumer genetic testing, what that might mean now that genetic DNA is everywhere, what the implications of that will be for forensic applications. So that's where we're headed. Okay, so how do we think about this sort of central ethical tension? I mean, what we're trying to do in forensic genetic testing, as Celeste has already so elegantly explained, is to basically protect the public by identifying people who are perpetrating sexual and violent crimes, right? People who are doing these crimes, we want them to not be doing those crimes. We want them to be behind bars, and engaging in this kind of testing of samples that are left at the scene of the crime is probably the best way of identifying people and getting them off the streets, right? And that's a clear, there's a clear, broad public interest in doing that. The primary ethical challenge, of course, though, in trying to apply these technologies is how to protect the public's interest in keeping perpetrators of sexual and violent crime off the street without unduly infringing the rights of individuals, many of whom may not have ever committed a crime and not really be suspects in crime, but might find themselves brought into forensic investigations as a consequence of some of these methodologies. How do we do this well? Until recently, it was possible to kind of think about this ethical challenge, about this sort of balancing of public protection versus individual interest, by basically keeping the kind of scope of forensic genetic profiling quite narrow. Keeping it narrowly based and focused on people who had previously committed crimes, having the methodologies of forensic genetic profiling being very carefully regulated and controlled. And that is still very much the case in most cases. But expanded genetic testing in the public domain and new law enforcement approaches, including these sort of genealogical approaches that Celeste already gave us an introduction to, are beginning to come in and seriously complicate the balance, because we're able to do things, ask questions, and basically bring under the veil of sort of genetic surveillance many more people than previously was the case. And so we have to be thinking very carefully about that balance between public protection and individual interest as we're thinking about these technologies. So, here's where I want to kind of segue to kind of thinking a little bit about sort of historically. In order to do this very intricate and wonderful comparison of a crime scene sample to a genetic profile of a potential criminal perpetrator, we need to basically have a database of genetic profiles against which to compare that crime scene sample. That was sort of implied in Celeste's comments, but she didn't really talk about that specifically. The database that was created originally on a national basis by the FBI to allow these kinds of comparisons and allow us to basically make identifications for crime scene suspects began with the DNA Identification Act of 1994 and was very specifically in the beginning narrowly focused on creating a national genetic profile database that would allow the FBI to track sex offenders. Over the course of time, there was gradual, basically as the methodology clearly showed promise, there was a gradual expansion in this database. First from sexual offenders to people who had committed violent crimes, people who had been convicted of felony offenses, to people who had, in certain states, who'd been convicted of misdemeanor offenses, to increasingly people who have just been arrested and accused of felony or misdemeanor offenses, but not necessarily convicted of those crimes. So that today we have a patchwork, basically, of policies of DNA collection in different states. Some states basically collect DNA only from individuals who've been convicted of felony crimes. Other states use a mix, have a mix of policies where state crime labs, for example, will collect DNA samples from all arrestees, but then the final profile will only go in upon conviction. In other cases, in many states, there's DNA collections just from felony arrests. And in many cases, people have the option of having their DNA profile removed from the DNA databases used for forensic genetic investigation if they are not subsequently convicted, but that is a complicated legal process and generally involves a lawyer. So basically, once your DNA is collected and your DNA profile of those 20 STRs is generated and goes into a database, it's very difficult to ultimately have your profile removed, whether or not you're convicted or not. This has allowed these databases to grow and to grow quite rapidly since 1994. They also mean that because of the ways in which individuals interact with the criminal justice system, and we know that people of color who are disproportionately interact with the criminal justice system, disproportionately are arrested, disproportionately serve time. We end up seeing more individuals of African American and Hispanic ethnicity, for example, overrepresented in these forensic genetic databases, with implications for who is more likely to then subsequently be matched in a subsequent criminal investigation. So there's already some potential justice concerns that relate to basically who is available for a match in the first place based on who comes into contact with the criminal justice system and whose DNA is present in these forensic genetic databases. This perhaps would be less of a concern. You might say, well, these individuals in most cases are people who have committed crimes and perhaps that still we're doing what we're supposed to be doing. But as was alluded to briefly, test reliability, although very, very good, can vary as a function of a bunch of things that are sometimes outside of the control of law enforcement. Sometimes there can be mixed samples left at crime scenes so that genetic profiles from multiple individuals, including the perpetrator and perhaps other people who might have just been in the vicinity, can basically contaminate the sample in various ways and make a profile more difficult to infer. In some cases, when there's very little genetic material, even with the intervention of the polymerase chain reaction, there can be degraded samples, which mean that we cannot get a good STR profile at every single one of those 20 loci, and so we end up with partial forensic genetic profiles, which can then be more difficult to interpret than complete profiles, perhaps more subject to potential inferential errors. And then we can also have, although great pains are taken to eliminate contamination, we can also have concerns with contamination by police or lab staff or other ways in which the crime scene can be contaminated, which can mean that someone who basically was not the perpetrator leaves their DNA, and that DNA is generated as part of the crime scene workup. So all of these things can affect the reliability of these tests, and because we have biased databases, this might mean that the potential for error might more differentially affect individuals from particular communities. Now, that's the downside of some of this. We also know that in some cases, partial genomic profiles in the forensic genetic context can actually be information that can be very useful and generative for our analyses. And this is particularly in the case, and as we were asking for examples earlier of why might, why do we want to look at more than one STR locus? Part of the reason is we can have these coincidental matches, but we might also have matching that has to do with relationships, family members that are closely genetically similar. And we've been able to take advantage of these partial genomic profiles as indications of relationship to make inferences about people who are not only within that federal database of offenders and arrestees for comparison, but might also give us clues to people who have not been arrested but might be close relatives. And this is a method known as familial DNA searching. Using an example, I borrowed one of Celeste's slides. So again, we have a crime scene sample with particular STR loci here. And in this case, we have one suspect who does not routinely and consistently match the crime scene sample and therefore can be excluded. But let's say we have another suspect here, a suspect who in certain cases is an exact match, a particular STR loci, but in all other cases, when there's not a complete match of both alleles, one allele is matching the crime scene sample in all cases in a manner that would be consistent with a close biological relationship. In this case, we could make a reliable inference that the individual who basically is matching the crime scene sample here is possibly the father or the son of the person who left the crime scene sample. So this kind of familial DNA searching, which can be done with various degrees of sort of statistical inference, is increasingly being used in a number of different states now, and in fact was successful in identifying one very infamous serial killer, Lonnie David Franklin Jr., the so-called Grim Sleeper killer, who had raped and murdered women in Southern California over a considerable period of time, almost 25 years, but with a big gap in between, which was how he came by the moniker sort of Grim Sleeper. There had been samples taken from the crime scene, no exact match in the California profile database, but there was a clear partial match, similar to that match that I showed on the previous slide, for an individual who turned out to be Franklin's son, Christopher, who had been previously convicted of a felony weapons charge and whose sample was therefore in the California offender database. On the basis of that match, the police decided that his father might be a lead suspect for these crimes. He, as in the case with Frank DeAngelo, was placed under surveillance, and actually an undercover policeman serving as a waiter was able to obtain his DNA from discarded pizza crusts at a pizza restaurant and confirm a match, and he was convicted as a consequence of these crimes. And so this familial matching has turned out to be an incredibly powerful additional tool to take advantage of these partial forensic genomic profiles. Now, identifying and correctly identifying partial genetic profile matches, though, is not nearly as definitive as that complete match. When we have partial matches, we can make inferences about degrees of relationship, but the assumptions that underlie our ability to make inferences about familial relationship are necessarily predicated in ways that are almost too complicated for me to explain, but I have a collaborator that I've worked with for a number of years who thinks about this work, that basically the ways that those STR varies in populations it influences our ability to make robust certain inferences about familial partial matches. And so we have to be much more careful when we're doing that kind of work. And as a consequence, my colleague, Rory Rolfs, who actually was a graduate student in biostatistics and genome sciences here at the University of Washington, when she was a student here, she began thinking about some of the assumptions that we make and some of the population background, genetic background assumptions that could influence our ability to make robust inferences, and we started thinking about the fact that errors could be made and we had to be very careful. And so we wrote a paper in 2013 basically saying, even as we're trying to use these techniques, we need to be much more transparent about what we're doing and the background assumptions that we're making to make sure that we're not differentially affecting groups from particular populations in ways that would be inappropriate. Now, a lot has moved and changed since 2013 when we first did that analysis and wrote that paper arguing for possible ethical concerns around familial matching. In particular, there has been an enormous explosion in public participation in direct-to-consumer genetic testing, both genetic testing for health and wellness related concerns, as well as in particular genetic ancestry testing. And this is a slide showing that many millions of people, particularly since 2017, have been engaging in direct-to-consumer genetic testing. And what was really shocking for me as I was putting my slides together, right now, as of 2018, it is estimated that approximately 12 million people in the United States have basically generated a genetic profile in the context of a direct-to-consumer genetic test, which is just about 4 million shy of the number of people that are currently contained in the National DNA Index Registry for forensic genetic testing. So very rapidly, the amount of genetic profiles that are present out there in the public is going to dwarf the amount of genetic information that we have present in these forensic genetic databases. So that's really interesting. In addition, and as Celeste began to sort of allude to, not only do we have many more samples and many more genetic profiles available potentially for forensic comparison, but we're actually assaying many more locations in the genome. Rather than looking at 20 simple tandem repeat regions, we're now assaying on average between sort of 500,000 and 1 million of these single nucleotide polymorphisms per genome. And these are being generated as a consequence of participation in things like 23andMe and Ancestry.com, and it's making that very detailed information, which allows us to robustly say things and matches about people, and also to make some very interesting long distance inferences about familial relationship, which have allowed identification of serial killers like the Golden State Killer case. Which was as a consequence of basically the police uploading a genotype array sample of several 100,000 SNPs into this GEDmatch database, a public genealogy database where people who had undergone direct-to-consumer genetic testing voluntarily placed their data into the public domain, and the police basically surreptitiously added their own sample here and said, are there any matches in the database? And with a match, and the match was to a bunch of like 15 or 20 third cousins within this database. And then the police, as Celeste has already described, basically had to trace back up to a common ancestor, a great-great-great-grandparent, and then trace back through the genealogies through thousands of individuals until they ended up with Frank DeAngelo. So it's a very powerful technique. Celeste mentioned that there was not just a single suspect, there was more than one. In fact, there were 2 false leads prior to identifying DeAngelo. And this is where this technique is very powerful, but it's sort of non-specific, because we basically have to kind of work up the genealogical tree and then work back down, and our inferences kind of get us in the right ballpark, but they don't get us to specificity. And one of the individuals who was sampled was an individual who was in a nursing home, who basically they had to get a subpoena to sample him. Another individual was surreptitiously followed around and sampled and was found not to be a match before we landed on Frank DeAngelo. And so I think there's some real concerns here about the technology and about its applications in this context. But those concerns notwithstanding, this technique is so powerful, and it's getting us to people that are in cold cases that have been unsolved for decades, that there's been an enormous uptick of interest. So the Golden State Killer case was solved, and basically a whole bunch of press was published about it in April of this year. In the interim, not 1, not 2, but 13 different cases have been solved using similar techniques. This is a table from a paper that was published last month in Science Magazine by Janov Ehrlich and colleagues. And they did an analysis of a set of samples from MyHeritage.com, an Ancestry direct-to-consumer genetic testing company, where they basically concluded that the ability to indirectly identify someone as a consequence of a match to a second or third cousin, that technique is going to be possible to implicate any individual in the US of European descent in the near future. Of the 1.2 million people who are already in heritage.com, 60% of those people could be identified by publicly available information right now. They were able to show as part of this analysis. So this has caused me and Rory, with whom I still collaborate, to kind of ask some really kind of complicated questions about what does this mean? I mean, people are putting their information in the public domain. It is implicating their relatives. Their relatives do not have a say. So decisions to place genetic information in public databases indirectly involve relatives, genealogies, family trees. And those other people don't get a say in whether they want their information to be rifled through with their awareness or consent. And basically making an argument, as we did earlier, for greater transparency, as well as for maybe to think carefully about the way that law enforcement interact with these public genetic databases. Should they be allowed to just simply upload a sample to a database for genealogy for law enforcement purposes? The GEDmatch system allows it, and the people who have their data there say that they're okay with having their data used in that way, but their relatives do not have a say. So maybe we need to be thinking more carefully about the regulations involved in some of this. Now, I'll be the first to admit that my opinions and my thoughts on this matter may not be the majority. This is a study that came out in early September in PLOS Biology by colleagues from Baylor University. Did a survey of 1,600 people around the United States, asking them what they thought about law enforcement, basically creating fake profiles and accessing genealogical websites for the purposes of forensic genetics searches, and very large majorities of the public thought that those kinds of law enforcement uses of genealogical information would be perfectly okay, particularly if they were in the service of trying to solve violent crimes or crimes involving children. So this is where this gets really interesting in terms of that kind of infringement of individual interests. I mean, where do we kind of basically say that people's individual interests in privacy get to be overridden if the hope and intent is that we're going to use that information to identify long-standing cold cases and identifying serial killers, for example. And just a final note, a paper that literally came out last week in Cell by Noah Rosenberg and colleagues, basically just showing that we can actually now cross-reference, so we basically can find individuals in forensic genetic databases for which we only have STR data, and we can cross-reference and find individuals that are likely to be their close relatives, who have SNP data but not STR data, and we can basically kind of cross-pollinate across databases and make inferences about the health-related implications of people in forensic genetic databases, or we can say things about the forensic linkages to people who are in research and commercial kind of health-related databases. So this is possible now with just a little bit of math. There are clearly consequences for the ways in which law enforcement are going to be able to sort of access genetic information, and there are also certainly consequences of genomic privacy for people who are participating in genomic research or placing their information in the public domain in order to say things about their health outcomes. They may also be exposing themselves to enhanced surveillance in various ways. And this is now possible because we can understand and assay genetic information so readily, and so much genetic information for so many people is available everywhere now, particularly in the United States, but increasingly globally. So, just a few things to chew on there. I went through it all very quickly, but just kind of in summary, to basically say that kind of the central concern here, the concern that we're going to continue to be animated about and be thinking about, I think, for a long time, because there are no easy answers, is how do we weigh the proportional infringement of private interests, interests in privacy, interests in our ability to sort of not be inappropriately surveilled against the public good? How do we think about that as a central ethical challenge? Acknowledging that there has been a pretty big mission creep with regard to forensic genetic testing, and it's only becoming more so with the broadening use of direct-to-consumer genetic testing. And that the best way to probably solve this problem is for law enforcement applications, and indeed any ways in which we are basically combining data, genetic data, across data sources, to do so with great transparency and with public discussion about what would be appropriate and what would not be so appropriate. So just a few links to sources here, some of the papers that I cited very quickly in passing if you want further information. This is very rapidly moving terrain, lots going on. And special thanks to Rory Rolls, who brought me to this topic and with whom I continue to collaborate. She's now a faculty member at San Francisco State University, as well as my colleague Lonnie Garrison, who is here in pediatrics here at the University of Washington. Thank you so much.